So we've already talked a little bit about protein synthesis. We've talked about the different cell organelles that are included. The instructions we've said are in little segments called genes that are located on the chromosomes. If we're looking at a eukaryotic cell, they're in the nucleus. If we're looking at a prokaryotic cell, there's only one chromosome and it's in that nucleoid. But either way, the DNA in those genes tells us the primary structure of the proteins, the order of the amino acids. And everything else, how the protein folds up into a three-dimensional shape, is really controlled by the order of the amino acids, by that primary structure. And you could think of the order of the amino acids in a protein like the letters that spell out a word. So if we have different amino acids, we get a different word, a different protein. If we have the same amino acids, but in a different order, we get a different protein. Just like if we have the same letters in a different order, we get a different word. If we have different letters, we get a different word. So tomorrow we'll talk a little bit about what you want to focus on for the test, and later today I'll have the study guide posted. So let's start out and look a little bit at RNA, because DNA may be the instructions to make the proteins, but the RNA is what actually reads those instructions and interprets them and builds the proteins. So we've already talked about DNA a little bit. So if we look at our picture, there's a DNA nucleotide on the right. And remember that one molecule of DNA is made of about 150 million nucleotides. And when we look at the DNA in a chromosome, it's not even one molecule. It's two molecules wrapped around each other in that double helix configuration. So we said when we look at a DNA nucleotide, all the nucleotides have deoxyribose sugar. All the nucleotides have a phosphate group. And then they have a nitrogenous base, something containing the amino or the amine functional group. And this nitrogenous base is either adenine, cytosine, guanine, or thymine. And usually we just abbreviate those with the letters. And as we'll see when we start looking at the genes, what really matters is the order of these letters. If we have the letters ACC, that might spell a different amino acid than if we have the letters AGT. So that's what's going to be our primary structure. And that's why we said a lot of times when we represent the DNA, we just completely leave out the deoxyribose sugar and the phosphate group because they're always the same. If we look at somebody with blue eyes and somebody with brown eyes, the difference isn't in their sugar. The difference isn't in their phosphate. The difference is in what's the order of these four letters in the gene that has the instructions for eye color. So we want to keep that in mind, that ultimately any genetic difference between two individuals can be reduced to what's the order of these four letters. Now, there can be environmental effects too. We talked about some of those the other day, but in terms of the biological difference, this is the biological difference, the order of those four letters. So let's compare a DNA nucleotide with an RNA nucleotide. And there are really two main differences. If we look at our RNA nucleotide, it has a phosphate group, just like the phosphate group in DNA. Chemically, no difference between them. If we look at the sugar, which is called a pentose sugar because it has 5 carbons, pent means 5, like a pentagram. DNA has deoxyribose sugar, that's where the D in the name comes from. RNA has ribose sugar, that's where the R in the name comes from. And DNA has ACG or T, RNA has ACG or U. And basically U has a very similar shape to T. Remember, at a molecular level, everything works according to its three-dimensional shape. So in RNA, we never see T's. In DNA, we never see U's. So if you have a strand of nucleotides, you can tell whether it's DNA or RNA by looking to see if there's T's in it or U's in it. Now, just like DNA, RNA is a long molecule. It's not as big as DNA is. DNA can be 150 million nucleotides bonded together. RNA is typically a few thousand, maybe only a few hundred. So it's not as big as DNA, but it's still a huge molecule. The other thing that's different is DNA, we have two molecules wrapped around each other in that double helix in the chromosomes. In RNA, there aren't two strands of RNA. There's just one strand of RNA. And usually it kind of flops around and forms what's usually called a cloverleaf shape, but you have to have a pretty vivid imagination to see a cloverleaf in the shape to tell the truth. So we want to keep in mind, in RNA, we're going to see U's instead of T's. Because again, with RNA, the ribose sugar is always the same in every nucleotide. The phosphate group is always the same in every nucleotide. So most of the time when we're looking at RNA, we're just going to leave the ribose out, and we're also going to leave the phosphate out. And we're just going to focus on that A, C, G, or U, because that's really where the information is. Now, when we look at RNA and we look at how RNA is made from the DNA, remember our rules for complementary base pairing. When we look at DNA, G and C always pair together, A and T always pair together. But when we make RNA from DNA, that's not going to work. If we have a G in the DNA, we're still going to get a C in the RNA. If we have a C, we're still going to get a G. If we have a T, we're still going to get an A. The difference is, if we have an A in our DNA, we can't get a T in the RNA, because remember, there is no T, no thymine in DNA. So instead, we're going to get a U, uracil. So this is a table for complementary base pairing between DNA and RNA. Now, if we had a similar table for complementary base pairing between DNA and DNA, it would look exactly the same, except there would be a T here. So we'll talk about this a little bit tomorrow, because later today we're going to find a third process or a third set of rules for complementary base pairing. So we talked about replication, and we explained how complementary base pairing explained how replication could occur very quickly and very accurately. So now what we want to think about is how we can make a protein. And making a protein occurs in two steps. And the first step is something called transcription. And in transcription, we're going to use our DNA to make messenger RNA. And there are lots of different types of messenger RNA. Messenger RNA, though, is used in translation. But whenever we make RNA in a cell, it's always made from DNA by transcription. So there's also something called rRNA in a cell, which is ribosomal RNA, and tRNA, which is transfer RNA, and snRNA, which is small nuclear RNA, and hRNA, which is heterogeneous RNA. So if we make DNA, if we take DNA, and we use it to make RNA, that's transcription. So that's going to be our first step in making a protein. Our second step in making a protein is translation. You have to be careful because these words both start with trans, but transcription is making RNA from DNA. Translation is making protein from the information that's on the messenger RNA. So the flow of information in living cells is usually represented as DNA makes RNA, makes protein. And notice here we're talking about an information flow. The DNA isn't converted to RNA, but the information on the DNA is used to build RNA. The RNA isn't converted to protein, but the information on the RNA is used to build protein. So let's look at transcription. And in some ways, transcription is similar to that process we talked about the other day, replication. So let's look at replication. So here's a section of a chromosome. We could be looking inside the nucleus of a eukaryotic cell, or we could be looking inside the nucleoid of a prokaryotic cell, a bacteria cell. So the kind of the sky blue represents our parental strands of DNA. Notice that one of the strands is called the template strand. This is the strand that has the pattern with the information that we're going to use to make a protein. So the template strand is the strand that we want to make messenger RNA from. So remember something we talked about with DNA. When we make new DNA or when we make RNA, we always start making it at the 5' end. Because since the RNA nucleotide looks similar to the DNA nucleotide, remember the end of the chain of nucleotides where the phosphate sticks out is the 5' end. So that means that we're looking at translation here, or transcription, I'm sorry. Transcription started here and it's gradually working over towards the right until we finish with this gene. So when we look at transcription, and we can see a small section along here, notice that if we look on our RNA, this purple strand, there's a U next to the A, there's C's next to G's, A's next to T's. So just like with the DNA, if we know the order of the nucleotides in the DNA, yesterday we said we could, or Thursday we said we could predict the order of the complementary strand of DNA. Notice here we can predict the order of the strand of RNA that gets made from it. So if we have a C on our DNA, we get a G in our RNA. If we have an A on our DNA, we get a U in our RNA. If we have a C, we get a G, and so on down the line. Now, notice that making RNA is controlled by an enzyme called RNA polymerase. So remember, we talked a little bit about DNA polymerase the other day. A polymerase makes a big molecule. Remember, ASE is always a hint that something is an enzyme. So DNA polymerase makes a big molecule of DNA. A polymer is a big molecule. RNA polymerase makes a big molecule of RNA. So whenever we make RNA from DNA, we need RNA polymerase. And basically, RNA polymerase does the job that all those different enzymes that we talked about during replication do. RNA polymerase splits the 2 strands of DNA in half. It adds new nucleotides. Now, where do we get these nucleotides? Well, if you're looking at a human cell, in a human cell, the place where we get these nucleotides is they're floating around in that nucleoplasm. They're kind of spare parts that the cell can use to build RNA. And they're just sitting around in the nucleoplasm, in the nucleus, waiting to get called. They're kind of like day workers. They're going to be used to make one particular protein, one particular molecule of RNA, which will be used to make a molecule of protein. Now, this is how we make all of our RNA. So we want to keep that in mind. We always make RNA by transcription. We always start at the 5' end of the new RNA and go to the 3' end. And we're going to focus on 3 types of RNA. If what we're making is rRNA, ribosomal RNA, then the cell's going to use this to build its ribosomes. And the rRNAs are actually going to start to be assembled into a ribosome in the nucleolus. They don't get completely built into a ribosome in the nucleolus. It's kind of like buying something at the store that's partially assembled and then you have to do some assembly when you get home. The rest of the nucleus is going to be built out in the cytoplasm. If we're making tRNA, which is transfer RNA, transfer RNA gets sent to the cytoplasm. And when the ribosomes start to make proteins, tRNA find the right amino acids and bring them to the ribosomes. So tRNA is one of the things that guarantees that that the amino acids get put into the correct order. The only type of RNA that we're going to actually use to make proteins in a cell is mRNA, which is messenger RNA. And in eukaryotic cells, the messenger RNA first is going to be transported out of the nucleus to a ribosome. And the messenger RNA, since it was transcribed off the DNA, this is going to carry the instructions for making our protein. So what are the instructions? Well, the instructions, remember what I said earlier, are going to be in the order of the letters. So when we make messenger RNA from DNA, the main thing that we have to remember is, what are our rules for complementary base pairing? And notice that we can also do the reverse if you think about it. If we have a piece of messenger RNA, we can actually build an artificial gene from it. Because if we have a section of RNA that's GUG, we know that the DNA that built that was CAC. And so what this allows us to do is, if we have a piece of messenger RNA, we can build an artificial gene. And then we can try to put that gene in a cell. That's one way that we do genetic engineering. Are there any questions about transcription before we look at the second half of translation? Okay, we're going to switch and we're going to look at translation now. So transcription was the process of making RNA from DNA. Translation is the process of using that information that's on the messenger RNA. Only messenger RNA gets translated. The other types of RNA just stay in the cell as RNA. So in translation, we're going to take messenger RNA and we're going to use that information to build a chromosome or a protein. The information was on the chromosome originally. So remember, the part of the cell that actually reads the instructions is called the ribosome. And ribosomes are made of two unequal halves. There's a large subunit and a small subunit. And normally they're close to each other, but they're not fully attached until a piece of messenger RNA arrives. And when the piece of messenger RNA arrives, they kind of clump together, and the messenger RNA is trapped in this little kind of groove in between them. So the messenger RNA sits there, and basically we're going to pull that messenger RNA through, kind of like if you've ever seen, you know, a ticker tape machine where the whatever gets pulled through. So we want to keep that in mind. They're going to come together at the start, which is always going to be the 5' end of the messenger RNA. And then they're going to move along the messenger RNA. And as they move along, the ribosome is going to go from the 5' end to the 3' end. Now, one of the things we want to keep in mind when a ribosome attaches to messenger RNA, remember we said RNA is a polymerase, it's a big molecule. We can have a ribosome attached to the 5' end, and when it gets down the line a little ways, another ribosome can attach to the 5' end. So we can actually use one piece of messenger RNA and we can make several proteins from it. And we can do it quickly because we can have several ribosomes attached to it at the same time. The second ribosome doesn't have to wait until the first ribosome is done. Now let's look at the real key to reading the message, the information that's on the messenger RNA. When we look at the ribosome looks at one codon at a time. A codon is 3 nucleotides. So that's a sugar connected to a phosphate, a sugar connected to a phosphate, a sugar connected to a phosphate, and then the 3 nucleotides, the A, C's, G's, are U's sticking out to the side. I said U, not T, because remember, this is RNA. So 3 nucleotides are the instructions for 1 amino acid. So notice one of the things this means is if we know how big our piece of messenger RNA is, if our messenger RNA is, say, 300 nucleotides long, if you think about it, if we have a piece of messenger RNA 300 nucleotides long, that means the biggest this protein can be is 100 amino acids, because it takes 3 nucleotides to give us the instructions for 1 amino acid. So if we know the size of the messenger RNA, we can figure out how big the protein could be. The reverse is also true. If you know that a protein has 6 amino acids, then it would have to have 6 times 3. There would have to be at least 18 nucleotides in the messenger RNA. So if we know how big the protein is, we can figure out the size of the messenger RNA. If we know how big the messenger RNA is, we can figure out the size of the protein. Now, there are 3 steps in translation. The first step is initiation, which is when the ribosome attaches to the messenger RNA. And elongation is adding amino acids to our protein that we're building. And then finally, termination happens when the messenger RNA is released from the ribosome. The ribosome lets go of it because the ribosome encounters something called a stop codon. So we'll talk more about codons in a moment, but let's look at what's happening at our ribosome. So here's a ribosome. This big yellow blob on the top is our large subunit. The littler yellow blob down on the bottom is the small subunit. This strip of yellow in between them represents our messenger RNA. Notice the 5' end is on the left, the 3' end is on the right. So that means this messenger RNA started, the ribosome started reading this messenger RNA on the left end, and it's gradually working to the right end. Now, if we look at our large subunit, it has 3 different sites. And if we look at these sites, this is kind of before, during, and after. So let's look up here at the top for a moment. We have these little kind of lime green boxes, and they've got these 3-letter, they're really abbreviations in them: ALA, SIR, HIS, GLU, GLY. These are the abbreviations for amino acids. There are 20 different amino acids in proteins. So let's look first at the P site. The P site, think of, remember, another name for a protein is a polypeptide. So think of protein or polypeptide. This is where we're adding an amino acid. So this green wormy looking thing is transfer RNA. We'll come back to this in a moment. But this transfer RNA has SER, serine, which is one of the amino acids in a protein, attached to it. And right now, this transfer RNA is adding serine to the protein that we're building. Over here in the E spot, we've got this blue wormy thing. Notice that this blue thing looks a little bit different than that green thing. This is transfer RNA, but there are different transfer RNAs and they all have slightly different shapes. This is the exit site. So when the ribosome moves, whatever's in the P site, the ribosome is going to move to the right. Whatever's in the P site will slide over into the E site. So notice this transfer RNA doesn't have a protein attached. This transfer RNA just a second ago added histidine. And now it doesn't have an amino acid, so it's going to exit. It's going to leave the ribosome. And then on the other side, we have this red worm. This is, again, a transfer RNA. This is the aminoacyl site. And this is where the next amino acid is going to come from. So this has an amino acid called ALA, alanine attached to it. And this is going to be the next amino acid we add after SEER, because when the ribosome moves down, it's going to move down one notch. So whatever is in the P site will go to E. Whatever is in the A site will go to P. Whatever is in the E site is going to get pushed out. What will go into the A site? Well, notice that the letters are color-coded down here. So CAC goes with the blue, UGA goes with the green, GCU goes with the red. So the ribosome is going to move down 3 letters. So then it'll be next to this GAG. That will be what's in the A site, the aminoacyl site. So the middle site is where we're adding an amino acid right now. The left side is where the transfer RNA is getting ready to leave because its amino acid is already part of the protein. And the right site, the A site, is the next transfer RNA that will add the next amino acid. Now, where do these transfer RNAs pick up the amino acids? Well, I said there were 20 amino acids, go way back to the beginning of the semester. And remember, one of the things we talked about was the cytoplasm is kind of a warehouse, and it stores lots of things. But one of the things that is stored in the cytoplasm is spare amino acids that can be used to build proteins. So transfer RNAs go out into the cytoplasm and they pick up an amino acid. Now, they don't pick up just any amino acid. If we had all of our transfer RNAs color-coded, this red transfer RNA would only pick up alanine, ALA. The green transfer RNA would only pick up serine. The blue one would only pick up histidine. And this is done because of, again, their chemistry, their 3-dimensional shape. Now, how do we get the right amino acid in the right spot? Well, that's where the codons on the messenger RNA come in. Notice that the codon that's in blue is CAC. If we look on the blue transfer RNA that's in the E side, notice that there are 3 letters here, G, U, G. Well, in RNA, G and C always pair together, and U and A always pair together. So notice that there's 3 nucleotides on our transfer RNA, and they're complementary to the 3 nucleotides in our codon. So because they fit up against the codon, they're called the anti-codon. Anti means against. So this means that this green transfer RNA that has the anticodon AGU can only attach to a piece of DNA that has the codon UCA. So UCA on the messenger RNA, this is the instructions for serine. And AGU will only pick up serine and will only fit in this spot. So it does happen very rarely, but it's almost impossible for the cell to make a mistake and put the wrong amino acid in the wrong spot. It can happen, but it's very, very rare. So let's look at these codons for a minute. Let's go down to a chart. So here's a chart we're going to see several times. We're using the same chart, but if you would go online and you would look up a chart, if you would do a search for a genetic code, you'll find a million different charts similar to this. Now we're going to focus on the inside. You can read the first letter in the codon over here, and the second letter up on top, and the third letter over to the right, but they have all 3 letters listed here. So suppose we have a codon that is UUU. Remember, codons are always 3 letters. Suppose we have a codon that is UUU. This is the codon on the messenger RNA for an amino acid that's abbreviated PHE. PHE is something called phenylalanine. Notice UUC is another codon, and it's also instructions for PHE. So when we look at the genetic code, there's a couple of things that we want to keep in mind. First of all, if we look at one codon like UUU, a codon only stands for one amino acid. A codon never stands for 2 amino acids because that would be ambiguous. Which amino acid would go into the protein? So if we have CCU, we're always going to put an amino acid abbreviated proline in that spot. If we have CAA, we're always going to put an amino acid that's abbreviated GLN, glycine, in that spot. Glutamine, sorry. So we want to keep that in mind. So one codon, one amino acid, with 3 exceptions. If we look up at UAA or UAG or UGA, if we look at these, notice they say stop. That's because when the ribosome hits these codons, this is the signal that tells the ribosome, don't add any more amino acids to this protein. This protein is done. So that's when that step that we talked about a minute ago, that's when termination occurs. The ribosome sees UAA on the messenger RNA, no more amino acids, so it lets go of the messenger RNA. This protein is done, it's finished. So we want to keep that in mind. Now notice another one of the amino acids, MET, is also labeled as start. So if you look at most of the messenger RNA that's made in a eukaryotic cell, like a human cell, like your cells, what we see is the first amino acid is almost always methionine, MET. So this means the beginning of the messenger RNA, the beginning of the instructions, is AUG. Now remember, one of the things we talked about earlier when we talked about what happens at the level of a cell, we said after the protein gets built, it travels inside the endoplasmic reticulum to the Golgi apparatus. And while it's in the ER, while it's in the Golgi apparatus, it gets modified. In most of our proteins, even though there's a MET amino acid at the beginning, at the start, later that MET gets cut out, it gets deleted. But there's a specially modified AUG, and that tells the amino acid, tells the ribosome, this is where you start building the protein. This is where you start adding amino acids. So we kind of have a start, and we have actually 3 different stops. So if we look at all the codons, most of the codons are the instructions for one amino acid. Now, notice that the genetic code is said to be redundant. Things that are redundant repeat themselves. So why do we say it's redundant? Well, because we have a lot of codons for the same amino acid. UUA is the codon for leucine. UUG is also the codon for leucine. CUU, CUC, CUA, CUG are all codons for leucine. So there's 20 different amino acids here. Some of them, like leucine, there's 6 different codons for leucine. On the other hand, for some of them, if we look, for tryptophan, TRP, there's only one codon, UGG. So the genetic code, each codon is one amino acid, but the code is redundant, which means there could be more than one codon for each amino acid. And there's a few of the codons, 3, that are stop codons and say, okay, this protein is over. Nothing else is going to go on here. So you might remember when we talked about replication, we talked about mutations. And we said a mutation is a change in the DNA. Well, think about if you have PHE, if you have this amino acid in a protein, you might have the codon UUU, so that means the amino acid was AAA. Well, think about what happens if you have a mutation that changes that amino acid to GAA. So instead of AAA, which would give us messenger RNA like this, suppose we have a mutation that changes the first A to G. So now we have GAA. So what type of RNA is complementary to GAA? Well, CUU. So notice instead of getting this amino acid, PHE, we would get a different one, leucine. So this is how mutations occur. So when you've heard about the variants of the COVID virus, those are caused by mutations. And if the mutations just change the DNA, so we go from AAA to AAG, notice that wouldn't matter because AAA, we'd get UUU for the messenger RNA. AAG, we'd get UUC for the messenger RNA, so we'd get the same amino acid. So nothing happens. But once we get a different amino acid, now a virus might be able to infect a new type of cell, or it might be more contagious, or it might be more deadly. Our immune system might have more difficulty in recognizing it and attacking it. For instance, the virus that causes the flu, that virus actually mutates very quickly. And that's why if you get the flu vaccine, that's why you have to get a different flu vaccine every year. Because if you get the flu vaccine this winter, by the time next winter comes around, the virus will have mutated. And what the vaccine does is it teaches your immune system to recognize the virus. So by the time next winter gets here, the virus is just enough different. Your immune system doesn't recognize it because the amino acids have changed because of mutations. And since the amino acids have changed, your immune system doesn't recognize it. So if you don't want to get the flu again, you might need to get a vaccine again. So we want to keep that in mind. So when we talk about mutations, if they change the amino acid, they can be important. But if they don't, they're not that big a deal. So let's look at one more screen. Let's look at a sample problem for the last homework set. So this is from protein synthesis problems. So let's work a sample. So we have our DNA template strand. So the first question is, how many amino acids can this section of DNA code for? So let's think about how we're going to figure out our answer. And our answer is going to be the number of nucleotides divided by 3. Because remember, it takes 3 nucleotides to make one codon, which is the instructions for one amino acid. So we've got 3, 6, 9, 12, 15, 18, if I'm counting right. So 6 is the maximum. Now, the reason I say 6 is the maximum, think about what we just discussed with the genetic code. With the genetic code, we could have a stop codon in there somewhere. So we might not even have 6, but that's the maximum we could get. Till we look it up on the chart and see exactly what's there, we wouldn't know. Okay, what strand of DNA is complementary to this? Okay, I'm going to come up here to make my DNA that's complementary. And then we'll cut and paste it and we'll move it down. Okay, so this is going to be 5 prime. Now notice, one of the key things when we're working these problems is, are we looking at 2 strands of DNA? Are we looking at 1 strand of DNA and 1 strand of RNA? Or are we looking at 2 strands of RNA, our messenger RNA and our transfer RNA? Well, this is what strand of DNA is complementary to the template. So this means the strand of DNA is going to be A, T, G, T, 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 G, oops, G, 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 C, 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 A, T, G, A. And then the 3' end is down here. So this is our complementary strand of DNA. So I'm going to write that down here. Write the messenger RNA that might be transcribed from this. Well, if we write the messenger RNA that comes from this, the messenger RNA is going to look almost like our DNA did. So it's going to be A, but now remember, RNA doesn't have a T, it doesn't have thymine. So what does A pair with? The U. So it's going to be AUG, UUU, GGG, CCC, AAA, and then UG, oops, that's not a U, and then UGA. And then again, we're going to have the 3' end down here. Now, let's compare our answer to number 2 and our answer to number 3. Because notice they're both complementary to our template strand. So if we look at number 2 and number 3, notice that the only difference between them is in number 2, wherever we see a T, in number 3, we see a U. Because wherever we see a T, that was something that was complementary to the A. And in RNA, it's U that's complementary to A. So if we look at the DNA that's complementary to our template strand, and if we look at the messenger RNA that we get from it, they're almost identical. We've got U's in the messenger RNA, we had T's. Okay, now write the amino acids that the messenger RNA will be translated to. Whenever it says write the amino acids, we're going to have to use that chart. So there's a chart down here at the end. So the first one was AUG, okay? That's met, our start. Okay, now we have sets of 3 letters at a time. So the first set of 3 letters after the start, after the met, is UUU. So that's going to be PHE. Then we have GGG. So if we look at GGG, that's GLY. So if we come back up here, okay, now we have 3 C's, now we have, and then after that we have 3 A's. So 3 C's is proline, 3 A's is lysine. And then we have UGA for our last 3 nucleotides. So if we go back and look that up, UGA is stop. Now, if we had some more messenger RNA, if we had some more DNA after this, notice it wouldn't matter for making the protein because once the ribosome sees a stop, it's over. Okay, now we're going to have a mutation to our template strand for number 5. So let's copy our DNA over. If a mutation to the template strand, I think it's supposed to say adds A to the strand between the 6th and 7th nucleotide, but I forgot to put that in there. So let's count over. So the 6th nucleotide is the A. So remember, the nucleotides are the letters. And really, it also includes the sugar and the phosphate they were attached to. Okay, so we're going to add another A here. So let's, oh, it did say. Okay, so now let's think about how we're going to figure out our new amino acids. First thing to keep in mind is you don't have to, but what some people find useful is to go into the messenger RNA or even the DNA. And since we're going to be looking at it 3 letters at a time, let's, oops. Let's highlight these. And I'm starting to run out of colors, so I'm just going to underline this last. Okay, so now when we look them up, it says write the new messenger RNA. So that was our DNA. So let's write our new messenger RNA. So again, keep in mind, we're going to read this 3 nucleotides at a time. I don't know why I keep changing the font color. I just want to change the fill. Okay. Now, some people just put a slash or something here. So let's look up our amino acids. Notice it's really asking us for 3 things in this problem. The new strand of DNA, the new template, the new messenger RNA. Well, notice AUG is exactly the same as it was before. UUU is exactly the same as it was before. So that means those are not going to change. So we've still got methionine, phenylalanine. Those are our first 2 amino acids. But now we have UGG. That's different. Notice before we had GGG in this spot. So let's look up UGG. So UGG is tryptophan, TRP. Now we have GCC. So if we look up GCC, that's alanine, ALA. Now we have CAA. And that's glutamine. And then finally, we have an AUG here. Well, AUG is MET. Notice that there can be MET amino acids in our protein. They don't have to only be at the start. Now notice we've got a T left over here at the end, except that T should be an A. I didn't convert it. So what happens? Well, notice here we hit a stop. So our protein was done. Here we haven't hit a stop yet. So we'll talk later, but what's going to happen with this A is the ribosome is going to keep reading 3 letters at a time until it hits a stop or until it runs out of messenger RNA. So that means this protein might end up being bigger than it's supposed to be. It's supposed to be 5 amino acids and then it stops. We've already got 6 and the ribosome is going to keep reading. So this is how we get mutations that produce proteins that don't work. Now, what type of mutation is this? Well, remember what we said earlier. If you have a mutation and you put an extra nucleotide in there, that's a type of mutation called an insertion mutation. And since all we did was add 1 mutation, one letter, that also means it's a point mutation. And notice how it throws everything out of whack after it occurs. Before we had 3 C's that we read together. Now we only have 2 C's that we read together. So when we have something like that, that's called a frame shift mutation, where it changes the kind of the beginning and end of those codons that we're looking at. So any questions about how we answer these types of questions? Okay, if not, I'm going to stop screen sharing. And if you don't have any questions, I will let you go now. This homework is down at the bottom of the content for this week. Later today, I'll post the study guide for the DNA test. And then tomorrow, we're going to talk a little bit about how cells control whether they make a protein or not, kind of the on-off switches, and also how we do genetic engineering, which is basically just changing the DNA of a cell. So we'll look at a few of those techniques. So if there are no questions, you can go ahead and go now, and we'll talk again tomorrow.